Hello students, I am Dr. Geetu Gambhir. Today I bring you the next learning episode in BSc Forensic Science on an important unit of the paper Technological Methods in Forensic Science, that is Electrophoresis. Dear students, in today's lecture you will be introduced to the technique of electrophoresis, its fundamental principle and methodology. You will also be told about the classification of electrophoretic techniques, common mediums used in electrophoresis, types of electrophoresis and factors affecting electrophoresis. We will wind up this module by studying about the application of electrophoresis in forensic science and conclusion. So students, let us have a look at the modules which we are going to learn today. In the module 1, an introduction to electrophoresis, module 2, fundamental principle and methodology, module 3, classification of electrophoretic technique, module 4, with the common mediums used in electrophoresis, and module 5, telling about the types of electrophoresis. Module 6 talks about the factors affecting electrophoresis, followed by application of electrophoresis in forensic science, and finally the conclusion in module 8. Introduction to electrophoresis. Electrophoresis may be defined as the migration of colloidal particles through a solution under influence of electrical field. Electrophoresis basically is the movement of distributed particles corresponding to the fluid under the influence of electric field. It mostly is known as electrokinetic phenomena. The technique of electrophoresis was discovered by Res in 1809 when he experimented that the soil particles dispersed in water migrate under the effect of an applied electric field. Electrophoresis take place because the particles dispersed in a fluid nearly at all times carry an electric surface charge. The charged molecules migrate to the oppositely charged electrodes, but that electric field is removed before it reaches there completely. Passage of charged particles in an electric field provides differential motion to the sample on the basis of charge and consequently resolves them. An electric field exerts electrostatic coulombic force on the particles through these charges. It has been witnessed that the colloidal particles are transferred to either the positive or negative electrode whenever a potential difference is applied between the two electrodes in a colloidal solution. Since electrophoresis is based on the differential movement of the electrically charged particles in an electric field, fundamentally the technique is appropriate only to ionic or ionogenic materials, that is, substances transformable to ionic species. Electrophoresis is recurrently used in forensic science, molecular biology and medicine. Fundamental principle and methodology. All types of electrophoresis are directed by the single set of general principles illustrated by equation. Mobility of a molecule is the ratio of the product of applied voltage and net charge on the molecule to the friction of the molecule. Mobility of the molecule or the migration rate increases with the increase in applied voltage and net charge on the molecule. On the other hand, the mobility of a molecule decreases with the increased molecular friction or resistance to movement through the viscous medium caused by the molecular size and shape. Overall, actual movement of the molecules increases with the increased time, since the movement is defined as the rate of migration. In a free buffer solution, in the absence of molecular sieving, the velocity v of a particle is proportional to the field strength E multiplied by its electrophoretic mobility mu. V is product of mu and E the field strength. The velocity v and the field strength E both are vectors, while the mobility mu is scalar, being positive for cations and negative for anions. The rate at which the migration takes place is dependent upon the strength of field, size and shape of the molecules, net charge, ionic strength, viscosity and temperature of the medium in which the molecules are moving. As an analytical technique, it is rapid, simple and very sensitive. Besides being a separation technique, it is also utilized diagnostically to study the properties of a single charged species. It provides the basis for various analytical techniques employed in separation of molecules by charge, size or binding affinity. The charged molecules under the stimulus of electric field travel in the direction of oppositely charged electrodes. Those molecules which are positively charged moves towards the cathode and negatively charged molecules move towards the anode. The charge on the molecules and the potential applied through the electrodes is responsible for this movement. The specimen under examination is placed at one end of the electrodes. The molecule starts moving towards the respective electrodes as soon as the electricity is applied. However, the molecular weight of the molecule influences this movement. Consequently, when mixture is placed on the electrophoresis paper or gel, different bands are observed along the paper after the process. This is a result of differential rate of migration by molecules based on their weight. Classification of electrophoretic techniques. Electrophoresis is commonly categorized on the basis of the existence or absence of a solid supporting medium or matrix by means of which the charged molecules travel in the electrophoretic system. Solution electrophoresis system utilizes aqueous buffers in the lack of presence of a solid support medium. Such systems can suffer by sample mixing caused by the diffusion of charged molecules with consequential loss of resolution during the sample application, separation and removal phases. Therefore, the solution electrophoresis systems must employ specific resources for stabilizing the aqueous solution in the electrophoresis cell. Generally, electrophoresis is classified on the basis of two criteria: initial component distribution and boundary permeability. Electrophoretic technique can be classified into four main types: zone electrophoresis represented as ZE, moving bound electrophoresis called as MBE, isotactophoresis ITP and isoelectric focusing IEF. Common mediums used in electrophoresis. Cellulose acetate. Cellulose acetate is a cellulosive derivative in which each hexose ring of the polysaccharide chain has two hydroxyl groups esterified to acetate, in general in C3 and C6 positions. Cellulose acetate is quite prevalent in clinical chemistry as it offers a convenient support that can be equilibrated with buffer in few seconds and yields good separations of proteins from biological fluids. Fully automatic systems have been manufactured around so that this system has become the principal model of the combined electrophoresis and robotics. Cellulose acetate allows the migration of even large serum proteins. However, the use of cellular acetate is limited to clinical electrophoresis and is not much in trend in basic research for high resolution runs, despite the fact that focusing and two-dimensional techniques on cellulose acetate membranes have been described. Agarose Gels. Agarose is a polysaccharide taken out of the seaweed. Agarose is purified linear galactane hydrocolloid extracted from agar or recuperated directly from agar bearing marine algae like Rhydophyta. Agarose gels are extremely easy to prepare and also non-toxic. Agarose gels have an extensive range of separation but comparatively low resolving power. Polyacrylamide gels. The most resourceful of all matrices are the polyacrylamide gels. Their popularity is because of the several fundamental properties: optical clarity, including ultraviolet to the range, the absence of the charged groups, and availability in wide range of pore sizes. Polyacrylamide is a cross-linked polymer of acrylamide. Polyacrylamide gels are considerably more exasperating to formulate than agarose gels. These gels have comparatively small range of separation but a very high resolving power. For characterization and separation of mixtures of proteins, these gels are used. Acrylamide is known potent neurotoxin. Types of electrophoresis. Affinity electrophoresis. The procedure consists of charge shift electrophoresis, mobility shift electrophoresis and affinity capillary electrophoresis. The techniques are established on the changes in electrophoretic pattern of molecules through biospecific interaction or complex formation. The interaction or binding of molecule, charged or uncharged, usually changes the electrophoretic properties of a molecule. Membrane proteins may be recognized by an alteration in mobility prompted by charged detergent. Nucleic acids or its fragments may be categorized by their affinity to other molecules. The technique have been used for assessment of binding constants. For example, in lectin affinity electrophoresis or characterization of molecules with particular characteristics like glycan content or ligand binding. Affinity electrophoresis may be used as an alternative quantification of the protein. Capillary electrophoresis. Capillary electrophoresis can be applied to separate ionic species by their charge and frictional forces and the hydrodynamic radius. In standard electrophoresis, electrically charged analytes migrate in conductive liquid medium under the impact of electric field. The technique of capillary electrophoresis was considered in 1960 to isolate species on the basis of their size to charge ratio inside the small capillary filled with an electrolyte. Immuno-electrophoresis. Immuno-electrophoresis is a common name for several biochemical methods used for isolation and characterization of proteins on the basis of their electrophoresis and reactions with antibodies. All types of immuno-electrophoresis need immunoglobulins, the antibodies which react with proteins, to be isolated or characterized. There are various types of immuno-electrophoresis. E-rocket immuno-electrophoresis is one of the one-dimensional quantitative immuno-electrophoresis. E-fused rocket immune-electrophoresis is an improvisation of one-dimensional quantitative immune electrophoresis utilized for thorough measurement of proteins in fraction from protein separation experiments. E-affinity immune electrophoresis is based on variation in the electrophoretic pattern of proteins through particular interaction or formation of complex with other macromolecules or ligands. Pulsed field gel electrophoresis. Pulsed field gel electrophoresis or PFGE is a system utilized for isolation of large deoxyribonucleic acid, that is DNA molecules, by application of an electric field to gel matrix that intermittently changes its course. Nonetheless, when field direction changes periodically, DNA have different lengths react at different rates to this change. Specifically, Large pieces of DNA will be slower to rearrange their charge when field direction is changed, while smaller pieces will be faster. Over the course of time, with the constant changing of the directions, each band will begin to separate gradually even at very large lengths. Thus, separation of very large DNA pieces using PFGE is made possible. Sodium dodecyl sulfate polyacrylamide gel electrophoresis. One of the conventional means of examining proteins by electrophoresis is by using sodium dodecyl sulfate polyacrylamide gel electrophoresis. SDS is a detergent which coats the linear protein sequence with the layer of SDS molecules and denatures it by binding it to the hydrophobic regions of the protein. Due to its negative charge, it becomes the dominant charge of the complex. The binding of number of SDS molecules is directly proportional to the size of the protein. Therefore, there should not be any change in charge to mass ratio with size. In solution, principally all different size proteins covered with SDS would run at about same mobility. However, the protein do not run through water. Instead, they run through an inert polymer known as polyacrylamide. Native gels. It is also possible to run protein gels without the SDS. These are known as native gels in that one does not deliberately denature the protein. Here, the native charge on the protein divided by its mass governs the movement of the protein in terms of the speed and direction. Electro-focusing gels. A different variant of gel electrophoresis is to decant a gel that purposefully has a pH gradient from one end to the other. The various ionizable groups either lose or pick up a proton as the protein is travelling through the pH gradient. Ultimately, it will find a pH where its charge is 0 and will get focused at that point. DNA agarose gels. The DNA agarose gel technique is simple process of separation of comparatively large DNA fragments from one another based on their size. The need for detergents is not there for DNA because it already consists of evenly spaced negative phosphate groups. Thus, as with SDS page, the charge to mass ratio is constant. Also, like SDS page, the separation results from the matrix itself. The range of size sensitivity can be varied by changing the density of agarose. DNA denaturing polyacrylamide gels are often called as sequencing gels. These gels usually contain additional denaturing compounds such as urea. Two pieces of DNA that differ in size by one base can be distinguished from each other in this way. Factors affecting electrophoresis. There are various factors which affect the technique of electrophoresis. For example, sample, electric field, buffer, supporting medium. Let us discuss them one by one. Sample. Charge. The net charge on the molecule affects the rate of migration. With increase in net charge, the rate of migration also increases. It is dependent on pH because the pH will determine the degree of ionization if it is a weak base or a weak acid. Size. Rate of migration is greater for the smaller molecules as compared to the bigger ones because of the decrease in frictional and electrostatic forces. Shape. Molecules with the similar charges but comprising of different shapes will affect the rate of migration. Electric field. According to Ohm's law, I is equal to V upon R, where I is the current, V the voltage and R the resistance. So, increase in current and voltage increases the migration rate, whereas increase in resistance will decrease the migration rate. Buffer. Buffer determines and stabilizes pH of the supporting medium. The composition of buffer, its ionic strength and the pH affects the rate of migration. In case of ionic strength of buffer, as the strength increases, the rate of migration decreases. Supporting medium. The sample molecules are retained on the supporting medium by the process of adsorption. The process of adsorption reduces the migration rate. Also, gels have sieve-like structures. The small pore size of sieves of the supporting medium hinders the movement of large molecules. Applications of electrophoresis in forensic science. The technique of electrophoresis has been employed in the field of forensics, as it can be used to isolate and compare DNA, blood proteins and inorganic constituents, for example, gunshot residues from scene of crime, suspects, victims or standard reference material. The technique of gel electrophoresis is used in forensics, molecular biology, genetics, microbiology and biochemistry. The results thereby obtained can be quantitatively analyzed by the visualization of gel with UV light and gel imaging devices. The image obtained from this step is recorded with the computer operated camera, and the intensity of the band or the spot of interest is measured and compared against the standard or markers loaded on the same gel. Conclusion. Electrophoresis basically is the movement of distributed particles corresponding to a fluid under the influence of electric field. Mobility of a molecule or the migration rate increases with increase in applied voltage and net charge on the molecule. Electrophoresis is commonly categorized on the basis of the existence or absence of a solid supporting medium or matrix by means of which the charged molecules travel in the electrophoretic system. The technique of electrophoresis has been employed in the field of forensic science. With all these informations here, we come to an end of today's lecture. Do keep in mind what we discussed today. I will be back with one more lecture in this series. If you want to learn more and enhance your knowledge, you may log on to our website www.cec.nic.in for MCQ, quizzes, LORs, etc. Thank you for your time today, and I will see you in the next lecture. Have a great day.